Well, welcome to Town Hall here in Barnstable. It's great to see you all. What a wonderful display here for the STEEP program. We want to thank certainly, University of Rhode Island and Harvard School of Public Health and Silent Spring for getting the grant you did and for going forward with this study. We've had the opportunity here in Barnstable. I'm Mark Ells, town manager. We've had the opportunity here in Barnstable to partner with Silent Spring for many years, looking at the many challenges that we have here on Cape Cod relevant to drinking water, safe drinking water, educating our public on what that means and how it's changing, and having certainly the support and the information that we gain by these sorts of efforts makes a huge difference for what we do here every day on the ground with our residents. So I can't thank you enough. We are active partners, supporters, so we're thrilled to see you here in Town Hall. I'm excited that you're going to go with our Water Division Supervisor this afternoon and tour our drinking water supply here in Hyannis. And anytime that we can partner, that we can support, we will do so here. So I'm very pleased to see you back here again today, participating, and I know they're filming this so we can share it beyond who's here today with our residents. I'll also, I have the privilege of knowing Cheryl Osimo, who has very graciously allowed me to get involved with all that you're doing. And so Cheryl plays multiple roles. She's a passionate individual who cares deeply for all that she does, but certainly in the areas as the co-founder of Silent Spring Institute, as the executive director of the Mass Breast Cancer Coalition. She plays a significant role in leading us toward better understanding of these challenges, and then how to actually get this information and fund this information and move forward in a positive direction. So I'm proud to say that I know and I'm a friend and a supporter of Cheryl. So Cheryl is your next speaker. Thank you.
Thank you very much, Mark. I appreciate that. Thank you, everyone, for coming to STEEP's first annual Science Day. I'm pleased that Cape residents will be able to hear from the whole STEEP team and to ask questions and share concerns, especially looking forward to hearing from the graduate students and postdocs. This is very, very exciting, who represent the next generation of scientists. A special round of applause for them for getting here today, bringing all this wonderful information. Thank you, and I can't wait to have a conversation with each and every one of you, so don't go home until we do, please. STEEP team is led by University of Rhode Island, together with Harvard T.H. Chan School of Public Health and Silent Spring Institute. And some of STEEP's work will be taking place here on Cape Cod. For our work on the Cape, we're partnering with Massachusetts Breast Cancer Coalition, Sierra Club Cape Cod Group, and others. Thank you to the volunteers that we have here today, and again to Mark Ells, who is a dear friend to all of us here on Cape Cod. He represents so many of us in many, many different ways. And he resides, by the way, right here in Centerville with his wife and beautiful 6 children. And again, a friend to all Cape citizens and always eager to help, especially in the environmental area. And I'd like to introduce his daughter, Elizabeth, who's home from college, a wonderful young lady following in her dad's footsteps. I know that to be true. Okay, so I'd like to now introduce Allison McCain and Laurel Shader, who are the leading community engagement activities here on Cape Cod as part of STEEP. Allison McCain is the Cooperative Extension Water Quality Coordinator in the College of Environmental Life Sciences and at the University of Rhode Island. Laurel Shader is a research scientist at Silent Spring Institute, where she leads the Institute's research on PFASs and other emerging drinking water contaminants. Laurel and Allison, thank you.
Good afternoon, everyone. Thank you so much for coming to the STEEP Center's first STEEP Science Day. Thank you to Cheryl for that great introduction and to Mark Ells for hosting us today. My name is Laurel Shader. I'm a research scientist at Silent Spring Institute, and I'm working together with Allison McCann from the University of Rhode Island, and we'll be leading the Cape Cod community engagement activities as part of the STEEP Center. As Cheryl mentioned, STEEP is a research study that is led by the University of Rhode Island, partnering with Harvard T.H. Chan School of Public Health and Silent Spring Institute. And for our Cape Cod activities, we'll be partnering with the Massachusetts Breast Cancer Coalition and the Sierra Club Cape Cod Group, as well as an advisory committee and many others in the community as well. This project is funded by the National Institute of Environmental Health Sciences. That's part of the National Institutes of Health. And I'm delighted that the whole STEEP team is here today. We're all wearing blue shirts, or almost all of us, so we're easy to find. And I'm really pleased that so many of the graduate students and postdocs are here, because you can hear straight from them about the work that they're doing as part of STEEP. And we'll have a poster session this afternoon as one of our activities, so you can ask a lot of questions. I also want to thank all the volunteers who are here today and have made the day run so smoothly. To the local businesses who donated food, and if you haven't eaten, please go to the conference room and get some food for this afternoon. And a special thanks to Hans Kaiser. Hans, can you wave hi? Hans is the superintendent of the Hyannis Water System, and he's graciously offered to host a tour so you can see some of this PFAS removal technology in action. So I've been on that tour a couple of times, and I hope that some of you will do that as well. And there's a map on both of the sign-in tables for that event. So our focus for today is on a class of chemicals called PFASs, or highly fluorinated chemicals. These are making headline news pretty much every day nowadays. There was an EPA summit a couple of weeks ago. There are communities all across the country that are discovering drinking water contamination, and pretty much every day there's something new about PFASs in the news. These are chemicals that are in all of us. They're in our stain-resistant carpets, they're in fast food packaging and non-stick pans. They're extremely persistent chemicals, and basically everybody is exposed to them. There are some communities that have higher levels of exposure because of drinking water contamination. Some of this contamination comes from the use of certain firefighting foams called AFFF. And Hyannis is one of the communities that has been addressing this type of pollution since 2015, when the levels of certain PFAS compounds were found above EPA health guidelines. And Hans and others in the town here have been working to put in treatment and to buy water from other towns and to address the source of the contamination. And PFASs have also shown up in other parts of the Cape, as we'll learn this afternoon. So our goals for today, and kind of our motto, is to ask, learn, and ask. Ask, learn, and act. That's a little hard to say. So we want you to ask questions. We want to hear your concerns. We want to hear from you and what you hope to hear from us as part of STEEP over the next 5 years of this project. We hope that you'll learn from our team about the different research projects, our community engagement work, and our research translation activities. And we hope that you'll act. We hope that you'll take the information that you learned today, share it with others, and continue to stay involved with STEEP. So right now we are in the part of our agenda with the STEEP project updates. So you'll hear from the different research and other components of the STEEP Center. We'll have a break at about 2:45. Before that, we'll have a Q&A session. So after each presentation, we'll try to make time for one quick question. But if you have a question, maybe jot it down, and we'll have plenty of time at the end for everyone to ask questions. And then at 3 o'clock, we'll have a few different activities that you can choose from. One will be an off-site tour of the Hyannis water system. One will be an opportunity here to mingle with the students. They have brought some of their work in poster form here, so you can look and ask questions about their work. And then we'll have a discussion in the conference room where all the food is to hear from you. Our research translation team would like to hear from you about what your concerns are and how you'd like to receive information about the STEEP Center. So with that, I'll hand it over to Allison, who will kick off this afternoon's activities.
Thanks, Laurel. Welcome, everybody. I do want to point out that on each one of the sign-in tables, there's an evaluation form. There's a few questions that we're asking. We wanted to get your input. What you thought about the day? How'd you hear about the event? What sections did you go to? What part of the day did you attend? And any comments and suggestions you have. We're looking for feedback so that we can help make this event as appropriate for Cape Cod community as we can in the future. So if you want to pick one of these up at some point and give us some feedback, just leave it on the sign-in tables. That would be fabulous. Really appreciate that. So with that, I have the pleasure of introducing our STEEP Superfund Research Program Program Director, Dr. Rainer Lohmann, who's professor at the University of Rhode Island Graduate School of Oceanography and has, at the beginning, has been our champion for moving herding cats, is often how it's referred to move our project forward, to get our project funded, and to keep us on track. It's a pleasure to introduce Rainer and welcome him to Science Day. Thanks, Rainer.
Thanks, Edison. Thanks, Laurel. Thanks, Mark. Thanks, Cheryl. Thank you, everybody. I would say cats and dogs comes more to mind, but anyhow. One quick word, so just so my parents are not in the room. They would have told me to dress formally, tie, suit, but I was told blue is the way to go today. So that's why we're all here in blue. So what I'm going to do is I'll give a 10-minute or so, a little bit of background, what has been happening with PFAS in the last year, and then we'll move into some of the specific what my group does, and you see some of very smart ladies in the back who wear blue, and they do great science, and I'll introduce what they do. So STEEP is an acronym, and it's really, we're looking at where the compounds are coming from, what are the sources, and of course drinking water is a big one, but there's more. There's what you get in your house, what you get through your diet. Transport, we're looking both in the groundwater of Cape Cod, but also longer and bigger distances. Of course, the human exposure part, and then what effects that has. And that's altogether the STEEP part. So it was mentioned a few times before, but we're looking at probably a few thousand sites in the US where communities have been or are exposed, most often because nearby was a fire training site, a DoD site, but also manufacturing of Teflon or other fluorinated chemicals. Airports, because of course they do work with firefighting foams. And more recently, like Hoosick Falls, there was manufacturing that used Teflon to make particular plastics with Teflon, so production sites too. On the general population level, it's some of the all-weather clothing you might use, so the outdoor jackets typically are treated with some fluorinated chemicals, which makes them great so you don't get wet, and even stains will run off. In the production of Teflon, these chemicals are used. Once it's in your frying pan, presumably everything is out. And of course, the stain-treated carpets and other things that you find potentially in this building, but certainly in hotels, major conference centers, schools, because they need to keep their carpets clean for a long time. So that's how we would be exposed to them on a daily basis. And that's also an exposure route for the Faroes people, for those that watch the film. Of course, the diet is a big one, but they also have a background exposure just like the rest of us. From a chemistry perspective, and you can forego the structures if you're afraid of chemistry, but they're fascinating chemicals because they have these ability to repel water and repel oil. That's very unique. That's why we use them so much. It turns out that in the food chain they work a little differently, unlike mercury or some of the other contaminants we're concerned about. So the food web is a little more difficult to assess, but nonetheless, if you eat too much whale, that's certainly not a good idea. And because we've had these chemicals produced for the last 50, 60 years, by now you have some ideas what effects you might be looking for. And the effects that you can find at background exposure, so any of us who are not exposed to high concentrations through either a particular diet or drinking water, and there you see some links to immunosuppression, and Philippe Grandjean has led a lot of that work. You see some links to metabolic disorders, insulin resistance, and so on, and potentially links to changes in your blood pressure. If you were really highly exposed, like next to a chemical manufacturing plant, now you look into several cancers that are linked to high exposure. So that's a very unique subgroup of people. And that has come out through numerous lawsuits, particularly of DuPont, because initially they didn't want to acknowledge that there was a problem. And then through a really smart lawyer, Rob Billett, he managed to get enough money of, I think it was a $4 million settlement initially, that he put into a big science study of 68,000 people who basically all donated blood. And then he could trace the history of exposure through atmosphere, water, and come up with plausible endpoints. So that's the high-end exposure to certain cancers. That was enough that finally EPA took notice and issued or worked with the manufacturers for a voluntary agreement to withdraw the two most worrisome compounds, PFOA and PFOS, and they are shown there in the chemical structures. So that was the good news, and we know that now if you look at human population, the concentration of blood are going down slowly. So the ban has had an effect, or the withdrawal has had an effect, but because we still want the chemistry in place, so industry has started replacing with other also fluorinated molecules. So we now go from a few compounds we know are of concern, and they're going down. Now we have others that might or might not be of concern. So that's kind of the landscape in which we're working right now. And that certainly makes research a little difficult because there's new compounds out there. As Laurel alluded, there has been a busy time, particularly in DC. So EPA hasn't done anything yet, but some of the states have. And you don't have to read all the entries, but New Jersey has been very active in issuing actually drinking water guidelines. And so they've come around, for PFOA, New Jersey is around 14, same for PFOS. If you live in Texas, you're really strongly built because you can withstand roughly 10 times that, which I guess is a good thing if you live in Texas. Vermont is similar to New Jersey, fairly strict. They're around 20 nanograms per liter. So I think by now, if you look at the states, you probably think a safe number might be in the low 20s. And there's rumor that EPA might have reached a similar conclusion. And so EPA had this secret summit that was only a few weeks ago, National Leadership Summit. And then there was a little problem because at the same time it came out that there was a study that EPA didn't want to have published because it's from another government agency, it's from the ATSDR, and presumably they would have suggested lowering the limit. So for now this is not been released, so we don't know. What everybody knows that the current levels are probably not safe. But then also turns out that to the summit you couldn't go if you came from a community because you were not invited. And if you were a reporter, you couldn't go to the summit either because you were thrown back out of the room. So it was a bit of a disaster. The good news is something's happened. Washington, Senator Shaheen was able to secure $7 million to look at a national health study linked to Pease Air Force Base. So there is some momentum coming, but it seems to be more from senators and congressmen rather than from the top down right now. Just to give you some idea, I was talking about nanograms per liter, and for you that doesn't necessarily mean much, but I want to give you some idea what we're talking about here. So that's, you get a nanogram if you take 10 grains of table salt and dissolve it in an Olympic-sized pool. So the concentration we measure are ridiculously small, but that is unfortunately enough to cause health effects. So what everybody's in the back showing you numbers is state-of-the-art instrumentation that measures very, very little, but still enough. All right, so steep. Everybody should be blue today, my mistake. But basically, what is so interesting and fascinating about this particular set of center is that we not only do fascinating science, which I hope we do, but also that we, as part of this construct, we have the community engagement part, which is why we're here today. We have the research translation, be that to towns, federal agencies, congressmen, whoever wants to hear from us. We have, of course, we train our grad students and postdocs, and we have something in place to actually run the whole thing. So it's a center, and it's meant to produce science that is actually relevant, which is, you know, somewhat nice. So what do we actually do? Well, we produce really pretty pictures, but they basically capture in that, and you can't read that, so I'll just skip over that, but it basically says we do science and we reach out to communities, to stakeholders, and so on. And then the other part we had to do is to make sure that the science projects, and you hear about them throughout the next few talks, that there's some link between them, and that was a very interesting and fascinating and somewhat tedious process to get different scientists to agree on a common set of goals. And so this was how we tried to make sure that the epidemiological informs what we do with a rodent model in the lab, which helps us understand the compounds we find in the environment, how they affect human health. So it's trying to go in a circle there. And so we were here before, and so I might start feeling at home now because we are in this room. We took a picture, and we looked just a little bit younger. Oops. Sorry, wrong picture. Ah, there we go. So we looked, but we had formal dresses, so today we're all in blue. There was one key person missing. It's Martha, who's running the admin call with me, so that's our team, and you see basically them all scattered around, and most of them, with one exception, wear blue. Well, I guess Clifton wears a bluish shirt. Kind of works. And Angela's shift. All right, so it's only you left, Clifton. Got to work on that. All right, so other than cracking bad jokes here, I'm also running a project and looking at how to detect these compounds. And so what we do is try to find out fairly simple ways of measuring these compounds. And one of them is what looks like that Halloween stuff. It's made of plastic. And so we try to use this particular plastic as a sampling mechanism to measure compounds that are in the atmosphere. So for example, we could try to use that to figure out whether in this particular town hall there's a high level of volatile fluorinated compounds. And who knows, we might even do that. Or in your home if you were interested. So that's how we try to use this thing, and then we do studies to see how the compounds get taken up by that sampler over time, and how we can use that to figure out what the concentrations are, either outdoor or indoors. Another, and I'm glad we talked about birds earlier on, is I have a grad student somewhere in the back who's hiding. Oh, no, she's not hiding. Hi, Anna. And so she's looking at these fluorinated compounds in birds. She's looking both at coastal birds from, I think it's in the Narragansett Bay, but so the North Atlantic seashore, and then those that are further off. And yes, but she only collects dead birds. So that's the good news. And then she cuts them into pieces. And then she realized that, of course, it shouldn't be a big surprise, but it's still always shocking when you realize that wildlife from anywhere is contaminated, be that near the coast or offshore. So, and there you see there's a bunch of contaminants in there. We're looking at maybe 24 compounds, and you find quite a few in different birds. And then part of her academic pursuit has to be able to figure out why do we see this, why are there these big changes, how much does it depend on what they eat, where they live, how much they migrate. And there's a poster somewhere that explains all of that. I think it's the one that's dripping with blood right now. The other thing we do is we try to come up with small tubes that help us measure what's in the water, maybe a pond, a river. And so there's another poster that has the, talking about a tube that's maybe that long. And we try to use that to measure the uptake of PFAS from water and potentially share that with maybe communities that are not nearby us. We can just ship them a small thing, put it in your water for a few weeks, ship it back to us, and we can tell you good, bad, or ugly, that kind of thing. And Christine has a poster on that somewhere in the back. There, yes. And then Jitka is going really small. I'm not sure she brought one of those fibers, but she has a fiber optic cable basically. So we're talking something that is, oh, she has one in her hand. Unfortunately, I've reached an age where I can't see that anymore. But I trust there is something. You look like you have really something. So it's a fiber that is really thin, and we hope to use that to figure out what is, to what degree are compounds dissolved, so we can measure them, or to what degree are they bound to sediment or soil. So we can only use the really contaminated sites, but we try to go to some sites where that's the case, and then use them to figure out how many of these compounds are mobile to leach out with the next rainstorm, and how many will just be stuck. In this case, stuck is a good thing. So, I thought I was trying to find a pretty picture. I'm not sure it is pretty, but basically what we're trying to do is, can we develop tools that measure what's in the atmosphere, and we could use them either inside or outside. We can use them that we can put in water, be that a river, lake, pond, coastal water, or something we can measure to what degree are the compounds that are present now in soil or sediment because of previous contamination, to what degree are they available to be taken up by biota, for example. And I think that's the end of my part. Oh yes, I've got to thank everybody. So, that was the end of my part.
Thanks, Reiner.
Thank you.
Is there a question for Reiner at this point? Yeah, please.
Yes, there's 6 of them, and I don't recall them right now, but I can find out. Yes, so they looked for, as part of a big episode, they looked for 35 cancerous endpoints, I believe. And for 6 of them, there's what they call a plausible link. Lawyers speak for, yes, you can sue the company for it. The other ones, there was no significant correlation.
Any other?
What depends what you mean by everywhere.
In the middle of the woods, with a pond in the middle of the woods, let's say?
Yes, it's both, yes. It is partially airborne. If you went to the South Pacific, we might be below detection limit. But in the Northern Hemisphere and the continental US, yeah, I think everywhere is a good description.
Thank you, Reiner. Reiner will be here too when we do the Q&A at the end of all of the updates. Our next speaker this afternoon is Cindy Hu. Cindy is with the Harvard T.H. Chan School of Public Health, Department of Environmental Health. And the title of her talk is PFASs, Where Are They From? How do they change in the environment, and how are humans exposed to them? Thank you, Cindy.
Thank you. Thank you very much for the nice introduction, Allison. I would like to thank each of you for coming here and for the opportunity to talk to you. I'll give you some updates about Project 1 of STEEP, which is actually led by my PhD advisor, Dr. Elsie Sunderland, who would love to be here today but couldn't due to some family emergencies. I want to answer these 3 big questions, but I only have 12 minutes, so let's just dive right in. Professor Lohmann already gave us a very nice overview about what these compounds are, and I would like to just reiterate a few key points to really make the message very substantial. So what are poly and perfluoroalkyl substances, or PFASs? They are a class of anthropogenic chemicals that are detected ubiquitously in American people, and data from the CDC suggests that 98% of American people are contaminated by these compounds. And they are very useful. They repel both water and oil. Therefore, you can find them in a wide variety of consumer products and industrial processes, such as rain jackets, pizza boxes, firefighting foams, carpet cleaners. And there was a question before about, can you detect these compounds everywhere in the environment? And the answer is almost yes, because these compounds are distributed globally. Scientists have found them in the most remote area on Earth, the Arctic. And the reason why they travel so far is there is this atmospheric pathway that they can take on long-range transport. And lastly, not only they are global, they also cause some severe local community concerns. And many communities across this country are struggling with this challenge of finding PFASs in our drinking water, and some of the potential health outcome or health concerns include immune system suppression and lipid metabolism disruption. So the first question we want to ask is where these compounds come from. And the technique we use here is called source attribution. This is a very useful technique because we will be able to look at multiple sources that contribute to PFAS contamination, compare them, and tell you which source is the most important one. And this is quite critical for environmental policy or environmental regulation, because if we want to reduce the exposure to these compounds, we need to find a very useful and important target of the major exposure source. These compounds are very complicated, but fortunately we are helped by two pieces of facts. One is PFAS is not one single compound. It's a class of diverse chemicals, and those chemicals are chemically related to each other. So when we compare multiple PFASs at the same time, if we found two or multiple of them are highly correlated with each other, that means that they're more likely to come from the same source, and this is a very useful information that we can derive from the environmental studies we do. And the second piece of fact that helped us is that there are some general rules about the physical chemical properties of these compounds. So by utilizing those rules, we can then make sense of the statistical results and then try to link that to the physical world. And to make this methods more concrete, I would like to summarize that to three major steps. The first step is we go out and we measure a wide variety of PFASs in the environment. And secondly, we use some multivariate advanced statistical tools to group them and look at the clustering of different PFAS compounds. And lastly is we use geospatial analysis or hydrological information to try to interpret the statistical results and make sense for the physical world. And let me give you an example of this study we did in 2014, where we collected around 30 surface water samples in Rhode Island and New York, and we measure a wide variety of PFAS compounds in them. And then we did this statistical analysis followed by hydrogeological analysis. And what we found is that when you look at multiple PFASs, they actually fall very nicely into three different groups. And one group of PFASs, we have found that they can be used as tracers for waste management sectors or landfills. And another group of PFASs we found can be used as tracers for metal plating industry. And this is very useful because then you can look at PFAS profile in the environmental samples and then come up with very important message of how regulators can reduce the contamination level or what source they should be go after. In this analysis, we are also doing some more work in New Hampshire near the single ban plastic factory to further refine this method and extend the list of PFASs we can measure in water samples. But the source is not the whole story, and now I'm moving to the second part of the talk, because the environment that PFASs are released into can also change them and change how they behave and change how they travel in the environment. And this kind of work, this part of the work is led by my colleague, Andrea Tokranov, and she has done her work right on the Cape at the Joint Base Cape Cod. So she has taken very detailed PFAS profiles in well water, and this is a map of the study site, where you can see that there are two sites that can cause potential contaminations. One is the fire training areas to the north, and then the second one is this wastewater infiltration bed to the south. And because we know the groundwater flow direction is from the north to south, then we can hypothesize that some of the wells that are more close to fire training areas and upstream to the wastewater infiltration bed are more impacted by the fire training area. And those wells to the south are more impacted by the wastewater infiltration bed. And her data support exactly this. And remember from the previous slides that I say different industrial sources are related to different PFASs. And here we can see that the elevation of one type of PFAS, PFOA, is associated more with the wastewater treatment plant release. And then the elevated percentage of PFAS is more related to the fire training area release. Another thing that makes PFAS is very hard to manage is the chemistry is quite complicated. There are some members in the PFAS family that are not stable by themselves, but then they can convert into more stable format. And once they complete this conversion, they will last in the environment for a really, really long time. And we call those compounds precursors. And precursors' behavior are very important to study because they can help us answer questions like, oh, we have two wells almost adjacent to each other, next to each other, but their PFAS concentrations are quite different. So we want to understand what is causing this difference, and studying precursors can tell us exactly that. Andrea is also extending her previous work to some ponds and lakes downstream from the fire training area, and we should expect more exciting data from her in the near future. Another very exciting work that is happening right on the Cape here is the Fish Mobile Laboratory, where scientists on our team expose fish to predetermined PFAS concentrations and look at how they bioaccumulate and how they affect the health of fish. And that can also have some implications about the human health concerns related to PFAS exposure. And then all the work we do is try to protect the public health and reduce exposure to these compounds. And this is also the last section of my talk, how are humans exposed to PFASs? I know here on the Cape, many residents are very concerned about the drinking water, but in terms of exposure pathways, there's more to that. And if you look at this graph, it may look very complicated in the beginning, but I would like to simplify it for you a little bit. On the top, we have this direct exposure route through consumer product usage. That means that when we use carbon cleaner or we eat food from this PFAS-containing food package food items, we can be exposed to them directly. But at the same time, there are also multiple pathways to the bottom part of this slide, and we call them environmental pathways. And this is because PFASs can be released into the environment during the entire product life cycle, and they eventually make their way into our water and our food, and humans can get exposed to them through those drinking water and marine food or seafood. The difference between the two groups, the top one and the bottom one, is that if I ban PFAS in consumer product today, then you should expect to this effect of lowering human exposure almost immediately. But because these compounds are very persistent in the environment, even after any regulatory actions, these compounds stick around in the environment, and they will continue to contribute to our exposure to PFASs. And to the back of the room, we have a poster on this study. This study is led by my colleague, Dr. Clifton Desensau, and he used birth cohort data from the Faroe Island, which is also highlighted in the movie you just saw this morning. And what he found was that since early 2000, after the phase out of some legacy PFASs in consumer product, the overall exposure level in Faroese children declined rapidly. But if you look at the section of the blue bar, that is a contribution from oil consumption. And this contribution stays relatively flat. I don't know if we have a pointer here, but if you can see my mouse, this blue section stays relatively flat, and that means that the relative importance of environmental sources is going to increase in the near future. And another study we have a poster to the back of the room on this. This is a study that is part of my PhD thesis, and I use very similar technique to the techniques I just talked about in the first part of this talk, this multivariate statistical analysis to look at human PFAS profile in serum and try to derive some useful information about exposure sources. And what we found was that some PFASs can be used as tracers for seafood exposure. Other PFASs can be used as tracers for consumer product exposure. So in the future, when we do community health studies, we can look at the PFAS profile in a community member's blood and then tell what is more likely to be the major exposure source of this person, so that we can give them advice on how to reduce their exposure. And that's the last substantial slide I have for the talk today. In order to reduce personal exposure, I would like to give everybody here some take-home messages. First is to figure out, is your drinking water contaminated if you're from this area of concern. And I know the community outreach core of STEEP is going to have a very detailed discussion on that, so I will just keep it brief. And another source is contaminated seafood or contaminated fish. So when you go out and fish in wild water bodies, it is important to be mindful of potential pollution in that route. And lastly, some consumer products may still contain PFASs. And one example I would like to give here is the microwave popcorn. So if anybody is a big fan of microwave popcorn, I would like to suggest to you stop eating that. Okay, that's all for what I have today. Thank you very much for your attention, and here are my acknowledgements. I would like to take a few questions if you have them. Thank you.
Are there some questions for Cindy? Yeah, David.
I was wondering, back in the mid-70s, I worked on a project in the freight and transport of PCBs.
Sure.
I was wondering if there's any lessons to be learned, since they're a complex group of chemicals. They're persistent and they bioaccumulate. If there's any lessons to be learned in how they convert the science of monitoring into health standards that could be used in the PFAS dialogue.
Yeah, thank you very much for the question. I'm very glad you mentioned PCB, because the technique we use for our project one, this multivariate statistical analysis, or we call them environmental forensics, is actually derived exactly based on studies done on PCBs and PAHs. And they're very similar to PFASs in the way that they are a class of chemicals and there are many members, and the interactions or also relationships among different members in that family can tell us a whole lot about where they're from and how have they traveled and etc. So yes, the study done on legacy compounds like PCBs can help us. But as Rainer has pointed out earlier in his talk, there are some unique properties of PFASs. This group of chemicals have very different properties than the traditional POPs or persistent organic pollutants. So we need to be mindful and accommodate that when we do our studies.
Thank you. We'll do one more, and then we'll take questions after.
Sure.
Yeah, I had a question. You had mentioned that they can stay persistent for long periods of time. Do you have like a range of what that would be, in time?
Yeah, so the carbon fluorine bound that is a signature of these compounds is the strongest in all organic chemistry. So people estimate if you wait for them to naturally break down, it's gonna take millennials. So fluorinated chemicals, there is another name called forever chemicals, and if you're interested, there are some op-eds online authored by Dr. Joel Allen, who is also on my PhD committee, and he has been calling them forever chemicals for a while.
Cindy, thank you very much.
Thank you very much.
Cindy will also be here during the question and answer discussion at the end of all of the updates. So if you still have a question, please write it down and note it. Talking to the mic. Oh, me? Oh, okay. Great. I thought my voice carried. Okay, thank you. Oh, okay, so our next speaker is Dr. Angela Slitt with the University of Rhode Island College of Pharmacy. And Angela is going to be talking about what makes PFASs unique. Thank you.
Good afternoon, everyone. It's nice to be back in Barnstable again, especially on such a lovely day like today. Just a few comments. So there was a question in the audience about fish, and what I would suggest to you is that the EPA does provide guidance on eating fish in general. And so if you just do a quick Google search for EPA PFOA fish, something like that, you can at least get to their health advisories regarding fish consumption and what's in fish and what's to eat and not. So that would be a good place to start. I teach freshmen sometimes, and that's one of the things we go over is where can you find good information about food consumption. Second of all, I just wanted to point out the popcorn. As a mom who's a Boy Scout mom, there's lots of different types of popcorn. So you can get some that are not in bags. Anyways, my sons would like that. All right, so let's talk a little bit about what makes PFAS unique. So our Project 3 is a combination between a chemical engineering program and lab. Dr. Bothan's lab is represented today. Jeff Bothan's not here, but he's a professor in chemical engineering. And then my laboratory is in the College of Pharmacy. And so it is kind of interesting because sometimes we have pharmacists and engineers in the room, and we definitely have learned we have different ways of thinking about things. But we jointly felt that it was a good opportunity to educate you about some of the qualities of PFASs that make them, as a class of chemicals, maybe more unique. So I will take a minute to talk about chemistry. We can't completely avoid it, but don't worry, this isn't going to be too complicated. So when we think about what some of our perfluorinated chemicals look like, and all of you sit there and look at the board, I think there's kind of one attribute that sticks out when you look at these chemical structures, right? So if we're looking, there's some carbons, but then what you're really noticing is that there are a lot of Fs. And what are those Fs? So those Fs are fluorine. And we notice between whether it's PFOA or PFAS or PFHXS, and these are just a few PFASs, that there are a lot of Fs. And one part about Fs is that fluorine is not really found for the most part in living systems. It's pretty rare. And so right away, that when we look at them from a chemical structural standpoint, they're unique. These are a very unique class of chemicals, and in one of our discussions back when we were writing the grants, one of the other members of the group, Bungsub Cho, said, wow, these are they're unlike anything you ever see because they have all these fluorines, and fluorines are so repellent. And for me as a scientist and more of a molecular biologist, biochemist, thought like, wow, yeah, how does our body kind of deal with the fact that they have something that's inherently repellent all of a sudden going into your system? And so this is something as a group of chemicals that we really appreciate, is that they structurally look very different, and that they're not something that our body necessarily has. But yet, with that said, they do resemble often what fats in our body. So on the left-hand side, maybe we can use the mouse. So on the left-hand side, we've got the structure for PFAS, PFOA. And again, the complicating thing with some of these is that even though they're both the same, they're really not. One of the PFOAs is a linear structure where the other is a branched. But what I wanted to illustrate on the right-hand side are these are common lipids, and these are the types of lipids that you would find in our body and in the membranes of our cells. And when you just kind of glance at them, we can kind of visually see that there are some similarities between the perfluorinated chemicals and fats in our body. So even though they have these unique fluorines that aren't found in nature, to some degree our body can, to some degree, seize them. They molecularly look a little bit like fats, and we know that these chemicals disturb fat homeostasis within rodents, and there's associations between disruptions with cholesterol in humans. So they can bind to fatty acid binding proteins and kind of leverage mechanisms in our cells that you utilize to transport lipids in and out. So as a group, our group in the project is kind of interested in that aspect of them, that even though they don't have a typical structure of chemical makeup that is something you'd find in your body, they do trick to some degree the body to act a lot like lipids that we do have present in our body. And so that's something we think about a lot. So as probably Reiner alluded to, is that they spend a long time in the body. And when we talk about time in the body from a pharmacokinetic standpoint, so being in a college of pharmacy, we think about things like drug half-life. If you take a drug, how long is it going to stay in your body before it's eliminated, and how often do you have to take a drug to keep it kind of working in the body? And so what's really kind of in particular unique to this class of chemicals, at least some of the PFASs, is that they have a very long half-life in the body. And to even define half-life, we're talking about how long it takes for half of something to go away. Sometimes you think about half-life when you hear about radiation or other types of things, but how long does it take? And in general, at least for PFAS and PFOA, and actually even for PFHXS, they take a long time for your body to get rid of half of it. The EPA numbers for PFAS are 5.4 years. For PFOA, it's 5.8 years. And I would just like to kind of give the example of something that's quite different. So if you take Tylenol to get rid of a headache, the half-life of Tylenol in your body is only a few hours, or opiates is only a few hours. And so even though we don't exactly know the chemical effects of all of these as a class of chemicals, there's some, as Reiner discussed, there's some kind of more well-described health effects that we know in work that Dr. Grandjean's done. We do know that half-life and the long half-life is something to be very concerned about when we put it in the perspective of other chemicals that we take on a daily basis that have a much shorter half-life. So we think about that a lot. And in the box, I just draw this kind of attention to some recent work that was just published in 2017. The numbers aren't exact, but in discussions with one of the community members here today, we were discussing well, what happens if you decide to stop drinking your water that may have some contamination? And this was a study that they did in Sweden, and they looked at half-life after the municipal water had been cleaned up and what the half-life looked like. And so you'll note that the half-lives were lower. They're still high, but they're a little bit lower than what the EPA reports. But again, these are still long. And the good thing is that people's levels went down over time. So that was very good, and we know that from other studies as well. So what do they do in the human body? Dr. Grandjean's group has spent a long time and have very interesting and seminal studies with antibody production for vaccines. There are other studies showing cancer effects, whether it's in Leydig cells, which are in the testes, or kidney cancer. Our group is interested in liver. We're liver biologists, and so we're interested in kind of following up on some of the liver effects that have been described in humans. So we do know that some of the PFASs are associated with some liver effects. They're very mild. And I don't really want to get into kind of the safety level of those, but just to say that the liver is a site at which these chemicals deposit, and there are associations between exposure and some of the changes one would see with changed liver function or changed injury. So the liver is not the only system, but it is one of the systems in the body that shows kind of what I refer to as a signal to PFAS exposure. And on the right, what we have is someone's liver, and for example, in the liver we have a protein called alanine aminotransferase, ALT. And if you go to the doctor and they draw your blood and maybe they'll do a Chem 16 panel, one of the things that they'll look for is, does ALT show up in your blood? So if your liver is slightly injured, and there's different ways that could happen. It's common, say, for example, sometimes with alcoholics, but sometimes if there's liver injury, the ALT leaves your liver. So this is showing ALT leaving your liver, and then when you take a blood draw, you can measure that ALT in the blood, and that's kind of, to some degree, reflective of liver injury. And we do know that there are some associations with PFOA and PFAS right now where we see slight increases in liver injury. This may not be the most important health effect of PFAS, so I want to make that clear. It's just it is a health effect that we see, and our group has the expertise to kind of ask other questions about liver effects. So that's part of what we do, and then we're bridging with our chemical engineers. And so our chemical engineers are more looking at physiochemical properties in liver, and then we'll integrate that with liver effects. So I put over here physio what? Because what are physiochemical properties? That's a long word. Physical chemical properties are how we describe the chemical substance. These physiochemical properties can dictate some of the properties that the chemical possesses inside a living system. And our projects seek to kind of look at PFAS as a group, understand their physiochemical properties, and then correlate them back to some of the effects that we're seeing in the lab with liver. So what are some physiochemical properties? And I come at this from the point of being in a college of pharmacy. So we kind of know that certain kind of characteristics about drugs may make them go to the urine or may make them get excreted through the feces. And we can utilize that information about the chemical structure to help inform us where the chemical might go or how it gets into cells. So one example would be molecular weight. So what is a molecular weight of a chemical? It's its composition, whether it has carbons or oxygens or nitrogens or hydrogens. It's how big or small the chemical is based on the elements that make it up. And I gave you 2 examples. So a large molecular weight chemical would be something like vancomycin, which some of you may have heard of. It's a very powerful antibiotic, and it's pretty big. Whereas something like Tylenol is a very small molecular weight. And PFAS is kind of more hang out towards the small molecular weight range. And why that matters is it helps us understand, again, how they may get into cells. What are some other properties? Some can be what we'd call lipophilicity or permeability. Lipophilicity has the root word lipo, thinking liposuction, right? Lipo is fat, but it's really referring to whether or not it's fat preferring. And so we ask the questions, does this chemical prefer to be in fat or in water? And that's kind of some of the things that Dr. Bothan's group and the chemical engineering group will kind of put together with the effects that we see in the lab. This really helps us understand if chemicals can penetrate cells. And if they're fat-loving, they're often permeable. So you might remember these types of experiments from when you were a kid, where you make these kind of clever things that look like waves. And we asked, do the chemicals go in the water portion, or do they go here in the oil portion? And this helps us understand how likely they are to deposit in certain areas of our body versus being eliminated. So how does all this relate to us and you? Well, for us, we'll try to better understand some of the properties of the PFASs, especially ones that are found in this area, that make them permeable to cells and relate them to the health effects that we see in the lab using liver tissues and liver cells and fat tissues. And then for you, what are you going to get out of this? Well, we hope that if we have more accurate values in some of these properties, we can help scientists, especially those at the EPA, better model using very complex computer programs what the exposure to humans and wildlife looks like. So what are we doing at URI right now? Right now we're doing work with liver cells. Emily's here. She has a poster and she's just done some really neat work with cultured liver cells from humans. And don't worry, we don't grab organs when people aren't looking or anything like that. These are hepatocytes that come from donors, and they're commercially available, and they're actually kind of combined from different donors into a single kind of what we call lot. And then we put them on culture dishes, and then we go ahead and do treatments of those, and then we take the materials, those kind of cells, once they're done being treated with the particular fluorinated chemical, and then ask questions about where their gene changes, where their protein changes. And so some of the ones, we're taking ones where some we know a lot about, such as PFOA and PFOS, and then ones we might know something about, and then some we may know very little about, and putting them in culture dishes, treating them in the lab, and then assessing the cell response. And I kind of have a mini Venn diagram here, because what we really want to do is be able to look at those physiochemical properties and ask, okay, what similarities do we see in the cell response? What differences do we see? Is there ways that we can sort these chemicals into more predictable types of behaviors? And on the bottom, this just shows kind of human hepatocyte, kind of how it starts. So the livers come from human donors, obviously with consent. These can be, sometimes we'll use normal liver tissue, sometimes we'll use hepatocytes from diseased livers. They get kind of isolated and digested, and then eventually they get stored in a way that they can come to us, and then we go ahead and thaw them and use them. It's actually a really powerful and great translational technique. So that's what we're in the thick of right now. I'm sure when we see you next year, we'll have data hopefully published by that point and give you much more information about that. along with some of the other work that we're doing in rodents and adipose tissue cells. I got it. Just want to acknowledge a few groups. First of all, the entire STEEP group, NIEHS, University of Rhode Island, Harvard, and then specifically my co-PI, Jeff Bothan, who's not here today, but we have a lot of fun together. My laboratory, this is a recent but obviously horrible picture. We needed to flip things around. We are better in the lab. And then the Bothan lab, who I got to hang out with them for a photo session, and we had a lot of fun. That was them teaching me how to do a selfie. Engineers learned a lot of fun. And with that, the only other thing I would say is the Research Translation Corps asked us to say sometimes about the things that we like to do when we're not doing science. And so my particular hobby is dog training, and this was me teaching my rescue Doberman how to use a treadmill, and somehow realizing maybe I needed to use the treadmill more than my dog. But anyways, with that, I'd like to take any questions, and thank you for your attention.
Very interesting. Are there any, is there any evidence of synergistic effects? So we oftentimes talk about one compound versus another, and we have numbers for each one, but is there a chance that it's the total load sometimes that can be a problem, even if all of them fall below some small number?
I think that's a really great question, and I think there are other examples from other classes of chemicals where that's seen. In fact, Philippe and I were just talking about that over lunch. And so one of the things that we're going to do as well is put combinations of these together. We actually had some recent conversations with the EPA, some EPA scientists last week about this, where they were specifically kind of asking us if we could propose to do that, where we can combine some of these at levels that normally are lower than the advisory level, but then ask the question is if when they're put together. So in our studies as we go forward, we will be kind of putting together mixtures and testing them at different concentrations to ask that question, because I think that's a really important question to address. Yes.
And we'll have more time for questions for Angie at the Q&A session at the end. All right, next up, we'll be hearing from the STEEP Community Engagement Corps, which is led by myself, and Allison McCann. So she'll get started, and then I'll pick up partway through.
Great, thank you. Okay, I just want to give a summary of the STEEP's Community Engagement Corps, working with our community partners to address community concerns, and listed here are our current community partners: Massachusetts Breast Cancer Coalition, the Sierra Club Cape Cod Group, and then we also work with a STEEP Cape Cod Advisory Committee, which is composed of representatives from Cape Cod. We meet quarterly, twice in person, and twice a year in person, and twice a year by phone call, and coordinate with all of these folks in terms of the work that the Community Engagement Corps is doing on the Cape. Just to summarize some of the activities, some of you may know one of the activities that we're most involved in right now is working to recruit volunteers for a private well testing program on Cape Cod to do some PFAS testing and then some report back. As I said, we're recruiting private well owners now. And we'll take those volunteers. We're looking to map those volunteers and then prioritize the testing. We're able to test 50 wells a year, and Laurel will talk more about that in her section of the update. But I wanted to just bring your attention to our flyer, which gives information about our private well testing, summarizes that, gives you, for those who are interested, where to go to volunteer, and then our contact information. So if you are a private well owner and you're interested, or you know private well owners who may be interested, there's flyers in the back of the room there for you to take and pass on. That would be fabulous. In addition to our private well testing and report back program, we're interested in looking at, among private well owners, what are the barriers and motivations that encourage or discourage private well owners to test their drinking water well. So we're going to be investigating through focus groups with private well owners in subsequent years their barriers and motivations to testing, to explore ways with these private well owners on how to promote testing of private wells to protect human health and to protect their families' health. We'll also be continuing to host this annual event throughout the course of STEEP, and looking forward to being involved in local community events as they evolve, and we can work to have information at these events, provide information to community groups at these events, and et cetera. This is a really simple diagram, but I think that it's a nice reminder. We've been talking in previous presentations about how we get exposed to PFASs, talking about drinking water on Cape Cod as one exposure route. Here's just a simple graphic of the water cycle, and then once we begin to dot the landscape, if you will, with land uses that we've spoken about earlier today, wastewater treatment, septic systems, areas where fire foams have been used, etc. You can start to see how the movement of water through the environment can also start to move chemicals through the environment. And as many of you may know, back in the dark ages, back in the 1980s, in the last century, Cape Cod was designated as a sole source aquifer. It was a popular program sponsored by the Environmental Protection Agency, and basically what it meant is, with that designation, that the groundwater on Cape Cod is the only source of drinking water for Cape Cod. No alternative exists. And therefore, it helped to, in one way, raise public attention that these areas afford our greatest attention for protection, and bring about an awareness around that. Sand and gravel aquifers, like the aquifers here on Cape Cod, are vulnerable to land use. They're unconfined from the surface, which means there's no confining layer that would prevent movement of contamination, if you will, down into the sands and gravels to get into the groundwater, where in areas we put public wells and private wells to draw the water out for our use. So it certainly affords our highest attention and protection. Courtesy of USGS, a publication from USGS, is this more specifically for Cape Cod, showing one of the 6 groundwater areas on the Cape. You can see here, with the help of my mouse, we have Nantucket Sound on the right, and then Cape Cod Bay here on the left. And what's interesting also in this slide is, in this graphic, is the interaction between groundwater and surface waters. So we can see that the water that falls to the earth moves down through the soil, gets recharged, recharges the groundwater, and with groundwater elevation and groundwater flow can discharge into surface water, as well as the surface water can move down through the soil and recharge the groundwater. And we heard today in various presentations about the interaction of groundwater and surface water and the importance of these PFASs as they move through the environment, both waterborne and airborne. Okay, Laurel. Laurel's going to talk a little bit more about our private well study, as well as some of the issues that have been found in Cape Cod drinking water.
Great, thanks, Allison. So there are a number of communities across the U.S. that are newly discovering PFAS contamination in their drinking water. PFAS have been identified as an issue on Cape Cod for a number of years now as part of EPA's testing program in 2013 to 2015 for unregulated contaminants, including 6 PFASs. The Hyannis Water System realized that 2 of their wells were above the EPA's health advisory at that time. And so those wells were taken offline and treatment was put in place to remove the PFAS and PFOA and get those levels below the guideline. And then in 2016, the EPA issued a new health advisory, a lifetime health advisory, which was much lower than their previous advisory. And so the Hyannis Water System had to take some extra steps to install new treatment and to purchase water to bring those levels below that new guideline. Hyannis is not the only area on Cape Cod, as we've heard about, that has PFAS contamination. There are those same firefighting foams that were used at the County Fire and Rescue Training area were also used at Joint Base Cape Cod. Cindy talked about some of the scientific research that our team is doing to understand how those chemicals move through the groundwater. Those chemicals have also migrated off-site and have ended up in some private wells, and so the Air Force is putting in treatment and providing water to homes where private wells have levels above that EPA guideline. In addition, there's a Mashpee well that's been taken offline, again, because of that foam contamination. In 2011, Silent Spring Institute conducted a study of private wells on Cape Cod. These were 20 wells located all throughout the Cape. We did have an emphasis on areas where we thought wells might be vulnerable to contamination. We weren't specifically focused on PFAS at that time. We were broadly interested in septic system impacts on water quality. So we were also looking for things like pharmaceuticals and flame retardants, and PFASs were one of the components of that study. We found 4 PFASs in at least half of the wells that we tested. These included some of the older legacy compounds like PFOS, which, as Philippe mentioned, 3M had phased out of production in the US by that time, as well as newer compounds like PFBS, which looks a whole lot like PFOS, it's just shorter, and that's what's now used in Scotchgard. So these replacement compounds were also common in the private wells that we tested. One of the things that we wanted to accomplish in our study was to identify the characteristics of vulnerable wells. Because this type of testing is expensive and hard to do in a routine fashion, we wanted to know whether wells that were showed signs of more septic system impact were more likely to have PFASs. And we found that to be the case. So for instance, wells with higher levels of nitrate tended to have more PFASs. And on Cape Cod, the largest source of nitrogen pollution into groundwater are septic systems. So it's consistent with septic systems being a source. And I should point out that the levels that we found here in the private wells were much lower and well below the health advisories, at least for what we have for PFOS and PFOA. But we did see that they were pretty common. And then we had a couple wells where the levels seemed much higher than we might have expected, just based on the nitrate alone. And it suggests there might be other sources. We know landfill leachate can contain PFASs. There are commercial businesses that have on-site wastewater. So there are other potential sources as well. So this was a study that we did in 2011. It was just with 20 wells. And so we're really glad to be a part of the STEEP Center and to be able to extend that work and to do additional testing of private wells on Cape Cod. Allison touched on some of the broad goals for our private well study. Our goal is to test for PFASs in Cape Cod private wells. This will help us improve our understanding of whether people are exposed to PFASs and at what level through their drinking water. It's going to help us identify the sources of PFAS contamination. Cindy talked a little bit about source identification and using different ratios of different chemicals to try to pin down, is this likely to be a foam source or a wastewater source, or what might the sources be? And so our work will feed into some of Dr. Sunderland's work. And then our study will also support testing of additional vulnerable wells in areas where they seem to be close to possible sources of contamination, and help homeowners identify treatment if necessary. So the scope of our project will include testing 50 wells each year of our study. So it's a 5-year project. So that's about 250 wells over the course of our study. As Allison mentioned, we are currently recruiting volunteers. So please feel free to visit the website to learn more and sign up if you're interested, or take some flyers and share them with your friends. And we anticipate collecting our first round of samples late in the summer. If you sign up, you'll be asked to fill out a consent form because it is a research study. And we'll also ask you some questions about your well, maybe if you have any nitrate data to help us get a sense of potential septic system impacts, the depth of your well, the age of your well. So we'll map out the locations for all the people who volunteer for our study, and we'll also plot out areas where there might be sources. Certainly we know where some of the foam sources are. We'll look at where landfills are. We'll look at areas with dense development that aren't served by sewer systems. But really we're hoping to learn more as well. We may not know what all the sources are. If you're a volunteer, you'll learn the results of your individual well water testing. At Silent Spring Institute, we have a tool called Derby that we've used for reporting results back to people of chemical testing in their household dust or in their urine. And it's a very helpful approach, especially for these types of chemicals where we really don't have a lot of benchmarks or a quote-unquote safe level that make for an easy interpretation. We let people see how they stack up to other people within the same study and provide information and steps that people can take in response to their results. Your own results will be kept confidential. We won't even let anyone know that you're in the study, but we will share study findings overall in public meetings like our annual update and in written reports as well. And in addition, the results will feed into Elsie Sunderland's work on the chemical fingerprinting. So with that, we look forward to continuing our work together. I hope we're staying on our own time schedule. No?
Yeah, we are.
We are, good.
We're good.
So with that, I guess we have time for a question or two. Yeah, David.
This is David Dow. I live in the Wacape Bay watershed in East Falmouth, which includes Mashpee, Joint Base Cape Cod, and the town of Sandwich. Of course, Joint Base Cape Cod spent over $1 billion studying the safe drinking water circular cleanup. We have expenditures for dealing with the nitrogen from our septic system, since apparently we're going to be sewered and have either community cluster system or new wastewater treatment plant out at the base. And of course, now we're learning about our PFAS problems both in Mashpee and Falmouth. And I was wondering if you have any advice about how the residents are going to pay for all this.
Boy, how will residents pay? Boy, that's a really good question. I don't know that I'm the one that has the answer to that question. I guess one thing that we've wanted towns to keep in mind as they think about how to deal with nitrogen pollution is to think about other sources of pollution, the other chemicals that are in wastewater. Wastewater is not the only source, or maybe the source for the highest levels of PFAS contamination, but we do know that wastewater contains other emerging contaminants. So we want to make sure whatever solution is adopted addresses both emerging contaminants as well as dealing with coastal water quality and nutrients. But as far as how to pay for it, I think that's perhaps not my area of expertise to answer.
I think it's another discussion, and I also think part of what we want to do with our focus groups for private well owners and on barriers and motivations to testing. I mean, one of the things we do know that private well owners, one of their barriers to test is the fact that they have to pay for that. And so how do we motivate, in the case of private well owners, how do we motivate private well owners to test, and what are the barriers, and how then as a community do we start to have the conversations of how do we pay for these things? So it's a good question. Yeah.
Okay, great. So for the last update for our STEEP team update, we'll hear from the Research Translation Corps. And just a few minutes ago, Philippe said that it's really important for the scientists to make sure that our science is informing policymakers and shaping policies that better protect public health. And so that's what our Research Translation team is here to do. We're hearing from Amber Neville, Nicole Rohr, and that's it. So take it away.
All right, thank you everybody for coming. As she said, this is Amber Neville and I'm Nicole Rohr, and the third person of our team who's not here right now is Judith Swift. And so we're going to show you some of the ways that you can keep informed on what STEEP is doing, perhaps find out more information on PFAS in general. And the first way is our website. So we have launched a website. It's uri.edu/STEEP, not too hard to remember. And we are also using traditional media, things like you may have seen some newspaper stories, some radio interviews, some different things like that. We also have social media. We use Twitter and Instagram and Facebook. You can find us on all of those. And we also do help out with the promotion for community events like today, and you may have seen some of our flyers or our social media cards announcing this event. So just a few things about the website, some things that we thought you guys might be interested in. When you visit our website, this is something that you will see, and you can find easily some recent news articles on PFAS, some recent events and highlights that the STEEP Center is doing, and then this middle panel is our Twitter feed, and so you can see what we've been tweeting about, who's been tweeting at us. We feature a lot of news stories, but also some work that the trainees and our researchers are doing. So from there, we have a whole page on Cape Cod, talks about your community in particular and things that you may be concerned about, specifically you know, the contamination, potential exposure through private well water, and also some of the other drinking water systems. We also have an page on the Faroe Islands if you are now intrigued to learn more about them. We have a tab on health risks, and so we will be expanding and updating and talking about health risks in general, as well as health risks that you might be concerned about here on the Cape. We talk about exposures. As you've heard numerous times today, the firefighting foams here on the Cape is a very large source of exposure for this community. Other communities have other risks, and so we talk a little bit about those. And specifically down here where you can find PFAS, these will have some tips about things that you may not be aware that you're being exposed to PFAS through and can work to reduce. Things like your waterproof, you know, outdoor gear and some things like that. You can find more information on each of the great research projects you heard about today, as well as the core work, so the Community Engagement Corps and the Research Translation Corps, and our Training Corps, which has all of our lovely trainees in it. The news, like we talked about, if you're just wanting to see what some of the latest big news articles are, you can come and see what we've highlighted. It might be a good place to start. And then you can always keep up with us on social media, which Amber's going to talk about a little bit more.
Great. So as Nicole said, we have our website and traditional media. We are also on Facebook, and you can get, you know, updates about our different events or what our trainees are doing out in the field, so you can get to know a little bit more about the actual work underway. We're on Twitter, where you can find both STEEP news, but we also share a lot of regional news and updates on our researchers out giving talks, like the one here of Philippe in the Faroe Islands. And we're also on Instagram, where we have where we're sharing, again, updates and pictures from the field. So we invite you to check out any of the social media channels that you use. And our goal, again, as the Research Translation Corps, is to provide you information in a variety of different methods. So we also have printed materials. So if you need some basic information about STEEP, we have our STEEP business card. I encourage you to grab a bookmark on the way out. It has all our different social media sites, and there's a basic one page about the STEEP project that you can find there too. Going forward, we're gonna have a series of short videos with the researchers, and also we'll be sharing some podcasts. We're gonna have a variety of infographics available on the website. So the idea is that, you know, we really are at the beginning stages, so we encourage you to keep checking back for more information. And we welcome all of your ideas and you telling us what information you need and the different ways you need it to be delivered to you. So we encourage you, if you can, to join us at 3 o'clock. We'll be in the side room, the one with the food, and we want to hear more feedback from everybody. We're also happy to take questions now if you have any. So I'll hand it back to.
Okay, so we have time now, about half an hour for a Q&A, so I'd invite all the speakers to come up to the front, and we have a microphone, so just please be sure to speak into the microphone so that it gets picked up on the recording, because this will be shown on TV so other people can watch as well.
Thank you.
I just wanted to say, if your question is for somebody specific that you know, then you could address that, or else we'll just pitch it to the team.
Right. Now, is there a technology available today to remove these particular chemicals from municipal water supplies?
Should we ask our resident expert in the audience? Because you are using some technology to remove it from the water supply. Remove PFAS.
The answer is yes.
There you go.
Do you want to say a little more about that, Hans?
We're going to do a tour of, Hans is actually hosting a tour at 3 o'clock.
In the Hyannis water system, we use activated carbon to remove the PFAS before that we have.
And I guess just to build on that answer, so activated carbon is good for removing PFAS and PFOA. There are some newer studies that suggest that activated carbon doesn't remove all PFASs. Again, it's a really diverse group of chemicals, and some of the shorter chain compounds seem to not be as well removed. So it depends a little bit on the specific chemical you're talking about.
Dr. Joyce Young, naturopathic physician. Since I'm particularly interested in fluorinated compounds in general, one of the questions that came to mind was, is the fluoride bond in all the PFAS, is it as tight in all of them? And what about other fluorinated compounds? Like I'm interested in fluoroquinolone drugs. And when I was here last time, I really thought about this issue of fluoroquinolone drugs and whether all your fluorinated drugs all have all your fluorinated compounds in general are really tightly bound and bonded.
I think I have the chances to go first. I think there's several pharmaceuticals where the CF3 group is added. And so it's, at that level, I guess the benefits outweigh the risk quite a bit. The chemicals that we're concerned of have longer chain and they're fully fluorinated, and at that stage they almost don't break down. You can tweak it with a lot of smart engineering, you can break them down, but that's very unusual situations. You can even use vitamin B12 catalyst to break them down, but that's nothing that would happen in the human body anytime soon. So I think when we talk about half-lives of a few years, we think of the time it takes to clean them back out of the system rather than break them down.
I'll add to that. So fluoroquinolones in particular, I think that their half-life is a lot faster than these, obviously. But interestingly, one of the designs that pharmaceutical companies have been kind of adopting more recently is this notion of fluorinating drugs, because it makes them resistant to metabolism. And a lot of the times when drugs are metabolized, there's often a potential for adverse drug effects because they get metabolized to things that can be toxic. So there's conversations, we've had conversations with this at scientific meetings, where the concern is now creating pharmaceuticals that are very resistant to degradation that people are eliminating, and then they're getting into the environment. So that it's a concern. So I can't think of specifically a new drug off of the top of my head that fits what you're talking about, but I do think that there's sometimes some concerns from maybe not ignorance per se, but one group having a motivation to create something safer for what they're doing and what works for them, but not really thinking about the consequences in another way. So you may find that there are other ones out there as pharmaceuticals. I can't think of ones right now, but I know there are.
Just a reminder, if you have to leave early, please do fill out the evaluation forms, because we really welcome your feedback on what you thought of today.
So just in general, has your group or anyone in the science community found specific levels in people's blood to be dangerous? And any examples of that?
Yeah, so I can cite one study from Dr. Philip Grandjean, where he used the children's blood data in Fair Island and look at the health outcome of immune suppression, and they have calculated some benchmark dose of pretty low level for PFOS and PFOA, as low as around 1 nanogram per ml found in children's blood. And that level is much lower than the average American people's blood according to NHANES data. So in terms of the level of concern, it's usually a function of what exactly the health outcome you are looking at. If you're looking at something very severe like cancer, it's probably going to be at a much higher level. But if you're looking at more subtle health effects like immune suppression, the level of concern can be quite low.
I should have clarified this, but specifically in certain areas, like, so say with Joint Base Cape Cod or like the Minnesota example, like those people who are getting really, really high concentrations of it, are you seeing cancer clusters around that? And is there any like hard data that I can access?
Yeah, I think if you look at the C8 health study, it is a very comprehensive epidemiological study down near West Virginia, where they have linked PFOA exposure to 6 diseases. And some of them, they do have the level of concern in terms of PFOA concentration in the blood. And cancer, I think I remember it being like bladder cancer or testicular cancer are 2 of the 6 diseases that scientists have found probable link between exposure and diseases. And some other ones are like lipid metabolism disruption and other diseases. So yeah, C8 health study.
Now, is there a requirement for municipal water suppliers to test their water systems for these chemicals? In other words, if I live in Philadelphia, does the Philadelphia Water Company have to make these tests?
I'm happy to jump in on that one. So as part of, PFASs are not currently regulated in drinking water. There's no federal drinking water standard and no requirement for routine monitoring or testing or meeting any kind of a guideline. There was a program from 2013 to 2015. It's called UCMR, the Unregulated Contaminant Monitoring Rule. It's part of the Safe Drinking Water Act. It goes in cycles, and EPA requires large public water supplies that serve at least 10,000 people and a handful of smaller supplies to test for chemicals that are on their radar screen, not currently regulated, but they want to get a sense of how common they are, because they won't regulate anything that's not very common. They haven't actually added any new drinking water standards in a really long time, but as part of their process, prevalence of chemicals is part of their criteria. So during that time, large public water supplies did have to do testing, and most, say here on the Cape, most of the public water supplies were large enough to fit in that category. But beyond that, there's no federal requirement for testing for PFASs. There are investigations underway, particularly around DoD sites right now, but in terms of a regular public water supply, no requirements.
Laurel, can I ask a follow-up question from the panel, because I don't know the answer either? If these, under this study, if they found elevated levels beyond the EPA advisory level of 70, were they obligated to make any kind of correction to eliminate them?
At a federal level, no. I think state agencies might have some authority to require the water supplies in their state to take an action. I know DEP was really involved here in Hyannis, but no. Short answer is no.
I just want to follow up, because I appreciate your question, but the question, how can we make it such that EPA does issue a mandatory drinking water guideline so that people have to test, or water systems have to be tested and potentially remediated? Because we think the evidence is strong enough that they cause a lot of concern and something has to change. That, unfortunately, is not a question that scientists can answer. It's a mostly political question at this point, and so there's different ways you can reach EPA, be that through a congressman and so on, or through public pressure, and I guess it would need all of that.
There was a letter from 25 senators asking, or that would, if it passed, it would require EPA to set a mandatory standard for PFAS and PFOA, but to my knowledge, that didn't kind of go too far.
I just wanted to add, too, that in addition to the federal standards, a state can adopt more stringent standards than federal guidelines. And so we've seen in a couple of different slide presentations this afternoon about states that have done some more stringent standards than the current health advisory level. So.
And for some states, those are a standard and enforceable, and sometimes the state doesn't have a mechanism to create its own enforceable standard, and those are more advisories or guidelines.
People talk about non-stick cookware like Teflon, but when you assess what people in general are using in terms of non-stick cookware, bakeware, and implements, there's really a huge wide range of these, a lot of them coming from China. And do you have any idea, because I'm really interested in, okay, where's the stuff coming from in order to advise patients? This is what the literature says, and this is what my recommendation is to you. Everybody wants to make their omelets in non-stick pans. So is there any information out there about all the other non-Teflon, non-stick cookware and bakeware? Almost all your bakeware right now is all non-stick too.
Start that and somebody can follow up, but I will say that the concern is not so much eating from a Teflon pan when it goes through the process of making it. So it's
Nicole?
not eating your omelet out of a Teflon pan.
Just use the mic.
So just all kinds of technical difficulties. So the concern is not so much eating your omelet out of a Teflon pan. If your Teflon gets really degraded and starts flaking, I think there's concerns. The main concern with Teflon is during the manufacturing process. So when you make the Teflon, when you apply it to the pan, so that's when you get sources of contamination at those manufacturing sites. But from the Teflon pan, it's not so much you getting it from when you make your omelet. But if you want to support these being used less, and you obviously don't want other people's communities to be impacted, it's those manufacturing sites. Now, as far as other kinds of pans, I don't know about that.
Do you know that if you overheat a Teflon pan, that those vapors, they know they're very harmful to birds? I can't imagine that they're only harmful to birds, but that's been the most well-documented toxicity that even the company mentions.
Any other comments on this particular question before we move on?
Well, it's just to back up what Nicole said. It's anecdotal, but a friend of mine works at the FDA, and they looked at is there a problem with Teflon exposure to these chemicals, and they basically scraped a Teflon pan into pieces and extracted it, and they couldn't get any of the P4 or PFOS out of it. It's basically gone by the time the Teflon pan is made. So that's the good news.
I had a question about autoimmune diseases. I've had Crohn's disease since my late 20s, and I know people have ulcerative colitis, and I remember that on the Parkersburg study, they suggested there was a relationship between the release of the PFOS, or I think it was PFOA, but anyway, and these autoimmune diseases in the large intestine. But the same is probably true of other autoimmune diseases, that most of them, they don't know how they're caused, and there's no treatment other than lifestyle changes. So I was wondering if there's any way to find out more definitively what the causes of these autoimmune diseases are and how to treat them.
Maybe Philippe might want to take a stab at that. He's hiding in the first row, but he's still here. Do you?
I mean, the bad thing is that as time goes by, we realize the problem is bigger than we thought. And so I've been arguing with EPA and ADSTR and with the European Food Safety Authority about where the safe limit is. And about 2008, so like 10 years ago, we thought that a water limit of several hundred ppt would be fine. And as we analyzed the data and we did some more studies, then we realized that was not quite sufficient. But in the beginning, we would look at some rodent studies. And then on the one hand, people would say, but people are not rodents. There must be a difference. And some sources from the private sector would say, oh, no, people are much more resistant. And so we had that argument going, and then when we finally began to see adverse effects in the general population, and children in particular, then the argument was, oh, but have you adjusted your findings for the father smoking, or, you know, something like that? Oh, you didn't? Okay, but, you know, that kind of argument. We need to know more about this before we say that these compounds are toxic to human health. And so this argument is still going on. To make a long story short, the limits have decreased, so currently the guideline, it's not a legal limit, the guideline from EPA is 7 APPT. We have done some calculations from our studies, and we believe that a water limit should be about 1 rather than 70. And so in comparison to several hundred, we're talking about orders of magnitude. And it's of course hard to believe that something can be so toxic that 1 part per trillion is toxic. But the point is that as you drink many liters of water during a year, you will eliminate the water again, but you will retain the PFAS is in your body, and they'll stay there for many years. And that's why we need to demand many fold lower levels in the contaminated water in order to protect health. So the battle is still going on. I know that ATSTR has this new version of the toxicology profile, which we haven't seen yet. I also know that the European Food Safety Authority has their report on its way. I think they finished it last fall. It's almost similar to the US situation. I don't know, but I believe that they're going to propose much lower limits than they did in 2008, and that maybe some discussions with the European Commission or national governments or something is going on, because with a very low limit, it's going to be an embarrassment to many countries, because they will suddenly find out something that was safe in 2008 is now a serious hazard. So I don't know where it's going to end up, but as researchers in the field, what we do is to try to generate the best possible information, even if that information may be unwanted for somebody responsible for the contaminated water or whatever it is. We do our best, and we try to keep those involved honest, and try to have them have the best possible information on their hands. And what the outcome is going to be is certainly something that we may all be capable of influencing one way or another, the way we vote, or which congressman we are calling or writing letters to. That's not a matter of science, unfortunately. We can't make that decision, but we can hold meetings like this, and we can speak up. And are you waving at me?
I just want to also follow up with that too. So just in general, immune disorders of the intestine are kind of on a population basis rising. And so we see, IBS more, Crohn's more. It's a complex issue to disentangle, because the American diet has also changed a lot over, the past few decades. But in general, it's really an understudied area compared to other types of systems within the body. So overall, I would say that's just an area that we know a lot less about, and there are fewer studies associating chemical exposure in general to those types of specific gut-related disorders. And then a challenge that we face in the laboratory is, unlike the fact that I was, teasingly saying that we could grab livers and, and culture them, there's a truth to the fact that complex immune disorders of the gut are very hard for us to model in the laboratory, because it requires a really kind of complex cell-based system. So it's an area that's understudied that needs to be advocated for, and there's a lot of work to be done in that area.
One more question, and then researchers will be here to interact with and things like that. Yep. Brief comment, please.
I used to take 500 milligrams tablets of sulfidine to control my Crohn's disease, but changing my lifestyle, I got down to the point now where I don't take any of them.
Thank you.
On the proactive side, have you done any research in terms of bottled water manufacturers and their interest in testing and disclosing this information to the public?
I'll take a first stab at that, and happy to hear from others as well. We haven't tested bottled water. My understanding is that bottled water falls under FDA, not EPA. And so unlike a public water supply that has to test for, not for PFAS, but for other things, there's really little testing and very little enforcement of bottled water. And even though your bottled water might look like it comes from somewhere pristine, it doesn't necessarily. It can just be tap water. So I'm not aware of other people that have done bottled water testing, but maybe others on our team. Oh. Yeah, I mean, that's a great question. I'm not aware of anyone who's tried to do that testing or any results. Has anyone else?
No, I'm not aware, but from everything I've gathered, I believe bottled water is hardly ever better than tap water. So I would be surprised if they were particularly keen on testing it for specific chemicals, because the odds are it's just going to be just as bad or worse than tap water.
Okay, so that brings to an end our formal Q&A session, but our team will be around for the rest of the afternoon. So we do have a bit of a break now until 3 o'clock. And then we have 3 different activities. So we'll be having a discussion with Nicole, Amber, and Judith in the small conference room where the food was. So maybe the food is still there so you can, This food is still there. And again, they'd like to hear from all of you about your questions and concerns and get some feedback about their communication strategies. In here, we'll have an informal discussion. The trainees will all be at the back of the room by their posters. So please feel free to ask them questions, and they'll be happy to talk about their work. And then again, Hans Kaiser, a big thanks to Hans for being willing to lead a tour. I've seen the water treatment a couple of times. It's one thing to look at a picture of it, but to see a big thing with 20 tons, is it 20 tons of activated carbon? Did I remember that? It's pretty impressive, and Hans does a great tour. So we have a map at both of the sign-in tables, or Allison is holding them in her hand, so please grab a map. I drew a little map, but you can also just type the address into your GPS. Oh, great. So perfect. So go see Hans over there by Allison if you're interested in that tour. And please fill out an evaluation form before you go. Okay, thank you, everyone.